Berti. It is too, you know, first, I love your energy. I love your colorful, you know, and the video you sent to us and we added in our LinkedIn and website really was great. Thanks for Andres also, Caicedo, for this animated and dynamic video. Thanks again. So, Stefania, you are assistant professor from Universidad de Concepción in Chile, lovely country. You obtain your PhD in immunology from King's College London, and then you have two postdoc at KCL, and then you move to your country in Chile to start your lab at the Universidad de Concepción. Your career was awarded by many awards as a female scientist, and last week I saw that you have also awarded something from one of people. So congratulations again for that. Your career, or you're actually working in mitochondrial transfer. You are with Andres, one of the pillar in this field, and we wish and we think that you will play a role in this very strategic hot topic today. You work too much in oral cancer, and in the dark side. Maybe it is not in the dark side as you said in the video, and the context of T helper response. Both of you, thank you very much for Andres and for Steffi. Volkmar, how can we work? We can start now for...
Yeah, sure. So Andre and Estefania,
please,
you take the...
Andres and Steffi, it's yours.
Yeah, you take the floor.
Excellent.
And David?
Well, thank you very much, Marvin, Volkmar, Stefania. It's really a pleasure, and I'm super proud to be here and to share some interesting insights and also be part of this great debate around what we should do with this horizontal mitochondrial transfer process. Don't you think, Steffi?
Yeah, of course. We have been very enthusiastic about promoting this symposium. We have been preparing like 9 slides for you. And of course, we would love to debate about the role of mitochondria in the transfer across cells.
Excellent. So I will start sharing my presentation. Hope that everybody is seeing it. Everything is okay. Can you see it?
Yes.
Perfect.
Yes.
Excellent. So I'm in charge to present a little intro, the big context, the dual role of this mitochondrial transfer. As many people know, I'm a big fan of Star Wars. Okay, so in Star Wars, George Lucas introduced the term midichlorians, inspired by what is mitochondria, and he did it quite well because he described it as microscopic life forms and introduced it to the public that without them, life would not exist. Well, eukaryotic, very complex life, I think, would not be able to exist. It's not moving. Let me see what is happening. Okay. So one particular aspect of these midichlorians is that whether Jedis or Siths that are aligned with evil, we could say it, as we posted in our video, both can harness these midichlorians mitochondrial powers to serve their own purposes. So maybe this is something that is happening with these mitochondria. Maybe mitochondria is, I don't know, maybe they are not bad, they are only good, but they are using and they are being used by the cells to their own purposes. And from reality now, we all agree that mitochondria responds to the cellular metabolic needs, to the state of differentiation, to the different types, and that mitochondria are critically important for normal and cancerous cells. So, well, actually, pretty much everybody knows, as we are in the World Mitochondria Society, that they were living free symbionts. Mitochondria evolved over time to share their DNA with the nucleus. And through sharing this DNA to the nucleus, they kind of lost their own capacity to be independently divided, but still they have very interesting aspects that have been investigated by different scientists. As I mentioned, they are different between all the tissues that we have, and they also change with age, But very interestingly, it has been observed that also our mental states maybe are changing some of the metabolic aspects of their function. And our great colleague, Martin Picard, he mentioned, and I like this concept, that he says that mitochondria are living, dynamic, they are maternally inherited, of course. They're energy transferring biosynthetic and signaling organelles, which is very interesting, and it goes with their role very well. But they also are part of this mitochondrial information processing system that he postulated, suggested in this publication, in which there is a crosstalk between the nucleus, the mitochondria, and other organelles. So interestingly, it's like if we are receiving mitochondria from other cell, what is happening to this mitochondrial information processing system? That's a very interesting information question, actually. And then we have Jonathan Brestow. Everybody knows his work, his amazing publications, and that he and a lot of very good scientists, some of them very good colleagues of mine, they introduced that intercellular and horizontal mitochondrial transfer is a process in which one or more mitochondria are transferred to a receptor cell. And in this publication, they dig and they observed that there was already some proof, some basic evidence of the mitochondrial transfer, some of that artificial, but also naturally occurring since 1979. So this is a very interesting and process that has already showed some evidence, and especially regarding the changes and modifications that can happen when a cell receives the extracellular mitochondria. And of course, my team that I like always to say hello and very thankful for their work. We were very interested in observing like mitochondria currently to Martin Picard, they're living, and maybe you can use that as drugs, living drugs, as for example CAR T cells, as we could be able to modify it, change it, and also it's very important to identify the good source. So these are important key questions. So the field of horizontal mitochondrial transfer is very active. So we have publications every day from now a lot of parts of the world. We have a publication from Turkey, from, for example, from the United States, from Asia, from everywhere, every part of the world, actually. And for example, we have good aspects. For example, the intercellular nanotube mediated mitochondrial transfer enhances the T cell metabolic fitness. Then the other side is that adipose stem cells transfer mitochondria to the cancer cells, inducing the resistance. And as we recently published with Dr. Berich and Jerry Nozzle, both amazing colleagues and scientists with an amazing team, that this process could be fundamental for the progression of cancer. And then we have also the basic roles, like it seems that, for example, stimulating cells support macrophage immunological homeostasis by transferring mitochondria. The work of Jonathan, in which he was able to transfer mitochondria to models of Leigh syndrome, observing that reduce mortality and morbidity, and that also, for example, there are some intentions to produce mitochondria are artificially in great quantity, but we still haven't arrived at, and we still use cells that produce a lot of mitochondria, for example, MCs, or could be any others. So this is very interesting. We have also a lot of great companies. We have Mitosense, we have Minovia, we have Selvi, and all of them have claimed amazing effects of this mitochondrial transfer. For example, they already have a patent submission for cells that have been enriched with mitochondria. There is a patent submission for the mitochondrial augmentation therapy, which is the transfer of mitochondria to these different cells. For example, from Selvi, they use already the basic concept that maybe mitochondria can be used to deliver specific factors such as mitochondrial DNA. With specific polymorphisms, for example, and that there is a need of composition to cryopreserve this fragile mitochondria that is outside the cells after we extract it artificially. Many things. So Recently, it was published in The Economist that, for example, mitochondrial transplants could cure diseases and lengthen lives. So it's a very promising concept. However, we still know, together with Stephanie and many others in the field, that we don't know if mitochondrial HMT is always a good thing. And if we stop HMT in cancer, maybe we could harm the homeostasis of other tissues. If we encourage or incentivize the horizontal mitochondrial transfer process, for example, in skin, can we increase the cancer risk? And what is the potential tumor risk when transplanting mitochondria, as some of companies are doing, like they are infusing mitochondria in patients, trying to observe what is happening, and also many other ways to administer mitochondria, for example, in the cephalorachidal liquid, in many other ways. So these are very important questions. So now that I've finished this small introduction, and as I will advocate for the good, I think that mitochondria is always a good thing, and we should move forward with the regenerative applications. What do you think, Steffi?
Yes. So, of course, you love to play the hero. I'm going to show you something not so good about mitochondria right now. Let me share my presentation, and you can tell me if you can see it.
Yes, everything fine.
Fine, okay. If something happened, let me know, because I just see my screen. Okay, guys, let's talk about mitochondrial transfer. First, I'm going to give a very brief overview about the nomenclature that Andres mentioned in the recent review from John and other important experts in the field. We're going to talk briefly as well about the mechanism. The function is going to go through Andres, because he loves to talk about the good side always of mitochondria. And I'm going to mention what I have been found in oral cancer in this aspect. So we have this consensus statement from John entitled Recommendation for Mitochondrial Transfer and Transplantation Nomenclature and Characterization. So here, John established some important terminology to talk about mitochondrial transfer. So we need to identify a donor cell, an acceptor cell, the cell that is going to give the mitochondria, and the cell is going to accept the mitochondria. And when this process happens, we have this process called horizontal mitochondrial transfer. In the case that the donor cell and the acceptor cells are known, we're going to call this mitochondrial transfer axis. And in the case that when we have one cell giving a load of mitochondria to different cells, or when the mitochondria is jumping from one cell that received the mitochondria to another cell, we're going to call this mitochondrial transfer network. How this mitochondria transfer between cells, it's important to understand that there have been defined two main mechanisms. One is contact dependent, and in the first one, I'm going to talk about the formation of nanotubes that allow the mitochondria to go from one cell to another, protected from the environment. And here I love to show these images because they're beautiful from Saha. And I love to show this as well because we have CD8 T cells, and I'm in immunology, so I love T cells. And how the cancer cells capture mitochondria from these T cells. Mitochondria, it's labeled with mitotracker green in green, and the nanotube, you can see it in red. When this process happens, we can observe high oxygen consumption rate, but when the nanotube formation is blocked through this chamber, this capacity is significantly reduced. And as well, when the authors did a knockdown on SIG3 and SIG5, proteins important for the formation of the nanotube, this mitochondrial transfer is as well significantly reduced. The second contact-dependent mechanism is the gap junction. So we have a protein very important here that is connecting 43. So here I can show you this lovely study showing the transfer of mitochondria between mesenchymal stroma cells and chondrocytes. So here they have the mitochondria in green from the mesenchymal stem cells and the chondrocytes in red, and they show how the connexin 43 staining enrich at the terminal ends of actin cellular extension containing mitochondria in the mesenchymal stem cells. Not only connexin 43, actin, and mitochondria is shown in these cells, they also show the formation of nanotubes, showing that it's not just one mechanism that happens when we are talking about mitochondrial transfer. So in here, what they did is they knocked down the expression of connexin 43 and truncate splicing protein as well. And in both cases, they see that a reduction of mitochondrial transfer. So they propose that the connexin 43 and the N-terminal truncated isoform, in this case GJA1, helps the mitochondria to be packaged in the vesicles or to move across the actin to promote this transfer through nanotubes or through vesicles. So we also have contact-independent mechanism, and here we have mitochondria traveling through vesicles. And this is an image from a review from John, but last year in the Congress in Berlin, I saw a very beautiful presentation from Dr. Pluchino, which was like amazing data showing mitochondrial transfer through extracellular vesicles between neural stem cells. So they did a proteomics of these EVs compared to the cell, and they saw that there was an enrichment of a lot of mitochondrial proteins in the EVs. They confirmed that as well with Western blot, and they checked as well DNA material from the mitochondria that was present when the EVs were there, but not DNA material from the nucleus, showing that the vesicles were protecting the mitochondria. Not only that, they also were able to show this beautiful image of expression of TOM20 with a technique called cryotem with nanogold antibodies, recognizing TOM20, showing that the intact mitochondria was there, but also they also analyzed CD63, showing the membrane of the vesicle and the mitochondria, the outer and the inner. So it was very nice to see not always the proteins, but also like the shape of the mitochondria within the vesicles. And this was going through macrophages, through endocytosis and phagocytosis, because when these processes were blocked or inhibited, the mitochondrial transfer was completely reduced, as you can see in the images here. So having mentioned already the main nomenclature associated with the terminology and the mechanism, John as well have a review showing like always that the mitochondria can improve like respiration, and it plays a good role in diseases, and Andres is going to talk about that. And in the review that he published like last month, you can see here the cancer cells that always, you can see these arrows, the cancer cell is always uptaking healthy mitochondria from other cells. So the mitochondria still is playing like a good role. Even if it's for the bad cell, that is a cancer cell, the mitochondria is doing a good thing, because for the cancer cell, it's going from a cell, this mitochondria, it's working properly, and it's going to help the cancer cell. So the mitochondria still is doing a good job. But what happened with the bad mitochondria here? And in the image, you have this white, can you see my cursor? Yeah, my arrow? No, maybe.
Yes, Steffi. Yeah, you can.
So what happened with this?
Thank you.
Yeah, what happened with this mitochondria in white that you can see in an arrow here going like out? But no one talks about what happened with the bad mitochondria, with the mitochondria that is dysfunctional, with the mitochondria that is not healthy. So in that context, this is what happened to me. So I'm gonna explain you why I have this issue about bad mitochondria, because I study cancer cells in the context of oral cancer, which is a cancer that affects mainly the mouth, is prevalent in male rather than female. It's increasing in men and in women over time. 90% of this cancer is manifested as oral squamous cell carcinoma. You can see in the map in blue the incidence, and in red the mortality of this cancer around the world. And the problem of this cancer is that it has a lot of pro-oxidative risk factors that affect directly the mouth, like smoking, which is very harmful for all of the cancer, for the health in general. But imagine how this affects directly the mouth. So it's very pro-oxidative risk factor, which is very commonly seen in the patients. They drink a lot, they have a alcoholic habit, they have bad nutrition habit, they have bad hygienic habit, and the microbiota has been related as well with this cancer lately. So what happened in this cancer in our lab? So we took biopsies from patients with oral cancer and biopsies from healthy controls. We put them in culture and we collect the secretome. So whatever the biopsy secrete, we took it and we performed proteomics. And this is when the mitochondria came to my life, because I love immunology. I love T cells, B cells, cytokines. Mitochondria was like completely new for me. But when I checked the pathways that were only present in cancer, I was very happy to see TCR, CD3, PD-1 signaling, which was related to exhaustion of T cells. But then I start to see all the pathways like crystal formation, mitochondrial protein import, respiratory electron transport, the Krebs cycle. And then I was a bit scared and I ignored the data. So then I checked the proteins that were shared between these two conditions, but because the abundance was different, they could have like different presence. And again, mitochondria, again, slapped my face, because a lot of proteins increase in cancer compared to healthy, related to mitochondrial structure and with the Krebs cycle. So after that, we run EPA analysis from Qiagen, and again, CT score, the highest CT score related to proteins associated with mitochondria processes, mitochondrial protein degradation, import, cellular response to hypoxia, and the ones that were downregulated were mitophagy and detoxification of ROS. And finally, when we check with the EPA again, disease and function, there were a lot of interesting function related to cancer, movement, migration, immunity, but I was very surprised about this one. There was tubulation of cells, which is related to the formation of nanotubes, which I show you at the beginning. That is one of the mechanisms for this. So then I was struggling a lot with this mitochondria data. I didn't know what to do, but I was lucky that one of my friends, an amazing scientist from Santiago, Universidad de los Andes, Patricia Luz, who by coincidence worked with Andrés in France, in Montpellier, she published a paper showing mitochondrial transfer between mesenchymal stem cells and T cells, showing that these mitochondria could improve OXPHOS and promote a regulatory phenotype. So I called Patty and I said, Patty, are you sure about this mitochondrial transfer thing? Because I'm going to write a grant about this. And she said, yeah, yes. Okay. So it was very important to try to understand whether the mitochondria could be transferring across cells and if this was important for this cancer. So we took, with the same EPA, we analyzed all of the pathways associated with mitochondrial transfer mechanism, like TNT, extracellular vesicle, cell junction, and you can see that in a triangle, circle, square, and organelle extraction that I didn't mention before, but a star. And again, look at all of the pathways that are increased in cancer compared to control related to mechanism of mitochondrial transfer. So our hypothesis was that metabolic reprogramming and exhaustion of effector T helper cells by tumor environment from oral cancer is mediated by mitochondrial transfer. Why? Because we observed that the T cells were there, they were expressing PD-1, which means that could become exhausted or it was the possibility that they become regulatory T cells as well, because we know that there's a lot of these cells in the cancer environment. And this was by a mechanism that I didn't know related to mitochondria. So here, Patty introduced me to Andres. Andres set up before and he published a very nice protocol called mitoception, in which we can isolate mitochondria, we can stain first, and we isolate mitochondria from different cells. In my case, I use a cell line. Ideally, I wanted to use biopsies to do this, but I started this project in 2020, so it was year of pandemia. So all the hospitals and university, everything was closed. So it was impossible to get biopsies at that period. So my option was to use a cell line. So we isolate, which I think was better for the standardization. We were able to isolate mitochondria. The T cells acquire these mitochondria, and we analyze proliferation, expression of exhaustion markers, and cytokine production. So here we can see the percentage of T cells that receive the mitochondria. They are mitochondrial positive. So we have 40% and above. We have cells that didn't receive mitochondria, and we have this in this yellow. We have the settings in which we did mitoception in those cells, but they didn't acquire the staining. So briefly, I'm not going to go through every day, but you can see that when the population here stain the T cells with citrus violet, when the population is moving to the left, the cell is proliferating more. So when you receive mitochondria from cancer cell, your proliferation is impaired. And this is significantly different between the cells that receive the mitochondria in comparison with the control. In terms of exhaustion markers, we can see that when the T cell acquired the mitochondria from cancer cell, increase the expression of PD-1, TIGIT, LAG3, PD-L1, showing that these cells start to become exhausted. Not for other markers, OX40, TIM3, CD25, and CTLA4 was so close to, it was significant, but it's so wishy-washy, but in the end, it was significant as well. Related to cytokine production, we can see that the acquisition of mitochondria from cancer reduced the production of interferon gamma, TNF, IL-4, IL-10, but it didn't affect IL-2 or IL-17. So all of this data is showing that basically when the T cell captured this mitochondria from the cancer cell, changed the phenotype, reduced the proliferation, and changed the cytokine secretion profile. So in our meetings in Chile, I met Keshav, and Keshav said it's very important to understand what is happening with the mitochondria, because he was studying mutations in mitochondria, and I completely agree with him. So I said, okay, let's go with proteomics and metabolomics to check what is happening with the cell when the cell captures these cancer mitochondria in comparison with the control cell. So we perform proteomics of the T cell that capture the mitochondria. We perform metabolomics in the same settings, but we also isolate mitochondria. We tried this with two different kits. One was better than the other one. And we check the proteomics of the mitochondria from the T cells and from the cancer cells to see if they have a different proteomic profile. So when we analyze, well, we did the analysis of the proteins, and then we finally present the pathways that were different between cells. And when we have a T cell that received the mitochondria from the cancer, we can see a pathway related with ROS production, hypoxia, and glycolysis. Whereas the T cells, the control T cells, express pathways associated with proliferation, cell cycling, and interferon gamma responses. Then when we move to isolated mitochondria and we check the pathways, again, we observe pathway related with ROS, glycolysis, and hypoxia. Whereas the mitochondria from the T cells, one of the highest pathway was oxidative phosphorylation, demonstrating that the metabolism of the cells is different, and the mitochondria is giving that profile. And one was preferred glycolysis, whereas the other one was more associated with OXPHOS. In the same setting here, we took the data and we performed IPA to analyze TOC list. And the highest talks that appear and was very significant here was mitochondrial dysfunction, saying that the mitochondria from the cancer cell is dysfunctional in comparison with the mitochondria from the T cell. Here you have all the proteins that validate this pathway, and in red you have the ones that were upregulated, in blue you have the ones that were downregulated in cancer compared to a T cell. So when we took these proteins and we check why the cancer cell has mitochondrial dysfunction, we have this image that I'm going to go through one by one. So first, the one that is upregulated is detoxification of ROS, meaning that there is production of ROS. Otherwise, why the mitochondria is going to have this pathway upregulated, the detoxification of ROS. Pathways that are downregulated, you can see it in blue, and the first one is related to aerobic respiration. So respiration of the mitochondria of the cancer cell is impaired. Why? We have problems with complex 3 assembly, complex 1 biogenesis, maturation of 3-carboxylic acid enzymes, and the regulation of the cycle, and we have reduction in the regulation of the pyruvate dehydrogenase. So when this enzyme is reduced, pyruvate has to go through this pathway through lactate and can't go or is reduced the movement through acetyl-CoA. Second pathway that was impaired in cancer mitochondria, mitophagy, which means that there's a problem with how these mitochondria that could be damaged is destroyed by the cell. And third, protein localization. Mitochondrial protein import was also reduced in mitochondria from cancer cells in comparison with T cells. So we understand a bit why mitochondria is defective in the cancer compared to a T cell, and why this could be causing problems in the T cell when the T cell acquires this mitochondria. Then, beside proteomics, we did metabolomics, which didn't give us a lot of results, but they were two very differentially expressed in both conditions, but it was reduced. They were both reduced in the cell that received mitochondria from cancer. And one is vitamin B1, or thymine. And probably you know that thymine is a cofactor of relevant enzyme of the 3-carboxylic acid cycle. Therefore, if this cofactor is reduced, we're going to have a problem in the entrance and the function of this cycle, and the cell is going to prefer to go through glycolysis. Is this correct? Can we confirm this? And we perform an experiment checking the uptake of glucose with the 2-MBDG, and here we add a new control that I didn't use it at the beginning because the cancer show cancer mitochondria or no mitochondria. But now we start to use autologous mitochondria in the mitoception to be sure that it was not the entrance of the mitochondria that was causing the problem. It was specifically a mitochondria from a cancer cell. So when we analyzed the glucose uptake, it was increased when we use mitochondria from cancer cells, but not autologous mitochondria or no mitochondria at all. Consumption of glucose was increased as well, and lactate as well. So because unfortunately I don't have a seahorse, but I have good friends in this meeting that we have in Chile about mitochondrial transfer, I met Rafael Arguello, a brilliant scientist, very clever, who developed a Zenit protocol to be able to analyze metabolic dependence in a single cell level, which allowed me to understand that the cancer cell was depending on glycolysis mainly, whereas the T cell was requiring glycolysis, but also the mitochondria. So we observe a difference in mitochondrial dependence that was high in the T cell in comparison with the cancer cell. However, the glycolytic capacity was higher in the cancer cell, showing that the metabolic profile of the cells is different, and that is mainly associated with the role of the mitochondria. What about ROS? What about the stress of the cells when they receive the mitochondria from the cancer cell? We observe that mitosocs in the CD4 T cells that acquire the mitochondria from a cancer cell induce more ROS and PD-1 expression, but not when you receive the autologous mitochondria, that you can see it by flow cytometry and confocal. So we finished with mitoception, and now we're optimizing natural mitochondrial transfer in the context of expression of PD-1, and then we're going to check ROS and so on. So we were able to do this in a contact-dependent mechanism and with a transwell system. And so far, it's working fine, but we need to stop more experiments for understanding like processes more associated now with mechanisms of mitochondrial transfer. So finally, as a cartoon, and for you to take this message, mitochondria is not always the angel. He is not always the good player. It could have dysfunctional effects in the cells that capture these mitochondria. And this could cause, in this case, exhaustion in my favorite cells, the T cells. So finally, I don't have as well time to go through this paper, but it was published very recently. It's a very good paper. I recommend you to read it. And it mentioned that here the problem of what happened when you transfer mitochondria that are not healthy. And they show that they transfer mitochondria, they transfer between a cancer cell into a T cell of mitochondria that has a problem with mitophagy. It can be degraded over time. The T cell is going to be full of these mitochondria, and the role of the T cell in the context of the anti-tumor response is going to be impaired. And just for you to know, we celebrate symposiums in Chile about mitochondrial transfer, and we finish the symposium with a party called MitoFest, like Oktoberfest. We do MitoFest. When we wear like white t-shirt, we draw mitochondria with fluorescence, and we have a lot of fun. And the first one, Andres was our barman. For the second symposium, we have one in Chile, and we have John Brestov, Keshav, and Rafael. With Patty, Andres, we start in Santiago, and then we move to Puerto Vara, south of Chile. Unfortunately, it was cloudy. You can't see the volcanoes, but it was very nice. And last year, half of the team, we have this mitochondria symposium in the north of Chile. So we're very active in promoting research and vinculation associated with mitochondrial function, mitochondrial transfer. Thank you very much for listening. And now we're going to see how Andres is going to convince you about the good side of mitochondria.
Amazing data. Steffi is a very compiling article. Congrats for all the work for you and all the team that is incredibly active over there in Concepcion. It's super cool, and I'm very proud to work with you and to collaborate with all our good friends and be the barman. I would like to prepare cocktails for everyone. So yeah, infusion. I put antioxidants as well in the cocktail. It's good. It's good to mitigate this stress response, so cells are not going to be exhausted. So that's super important. Which brings us a lot of questions. It brings us a lot of questions. I think that at the end, I think that everybody will have a lot of interesting. Yeah. So yes, don't forget to add your questions to the chat, and or dear representatives of Mitochondria Society, Volmar and Marvin will definitely pick up the ones that could be more interesting. But all questions are good. So now defending my position, which is going to be tough, but we will do our best to do that. I will start sharing my screen regarding the good. Okay, so regarding mitochondrial transfer, there are cells that are very supportive of tissue repair and regeneration. They can differentiate, and it has been observed by a lot of scientists, amazing colleagues everywhere, that, for example, mesenchymal stem cells have the capacity to transfer these mitochondria and to modulate the immune response. And as connections is super important, collaborative work is even more important when the science, as Steffi mentioned, we collaborate with Patty in one of these articles that show that mesenchymal stem cells are transferring mitochondria to the Th17 cells, and they're inducing the Tregs program. So, for example, here in this image, we are observing the MCs labeled with mitochondria mitotracker red, and they are transferring their mitochondria to these T cells. And it's very interesting to know that there's always a diverse populations that not always the same homogeneous population of cells that uptake mitochondria, but there are different ones. Maybe this could mean that they have different properties. We should try to dig in that question a little more. And we observe it, they are taking these mitochondria with the mitotracker red. And also to be sure, we sequence the mesenchymal stem cells mtDNA, and we observe how much time this mtDNA was inside the cells. And we observed that there was a decay after 72 hours. But between 4 and 24 hours, the quantity of mitochondria was quite strong in the cells that I've taken. Later, after putting them all together during 4 hours, we were able to observe that mitochondria that was transferred from mesenchymal stem cells to the Th17s lowered their pro-inflammatory profile and induced an immunoregulatory profile, and also increased the max respiration of the cells. And then after, when we transfer the mitochondria to these Th17 cells, we were able, that was a decrease in TNF and interferon gamma. So amazing article, and also that highlights the immunoregulatory capacity of these mesenchymal stem cells that transfer mitochondria to the pro-inflammatory cells, which is very important for tissue maintenance after damage, for example, and other immune diseases, for example, as has been recently published by Patty. Later after, we were able to observe by many others that, for example, the transfer of mitochondria from these mesenchymal stem cells mediate FOXP3 stability, and they observe as well as Steffi that this transfer is mediated by nanotubular structures, and that interferon gamma was lower and FOXP3 was more stable. So that's good. Then with the favorite cells of Steffi, the CD8 plus cells, we were able, this amazing team led by Javier Hernandez was able to observe that when mesenchymal stem cells were transferring the mitochondria to the CD8 plus cells, there was a decrease in number of CD8 T cells. So it is important, reinforcing the point of immune regulation, which is good for tissue maintenance. One of the key papers in the field that was published several years ago, they induce lung injury with LPS, and then they add mesenchymal stem cells. Amazing work by Mohan Islam and Yar Bhattacharya, in which they observed that the mesenchymal stem cells that arrive to the alveoli were transferring mitochondria, and this process was mediated by gap junctions. They observed that the supportive role of these mesenchymal stem cells in that environment, in that harm environment, help in the production of ATP, but also in the production of surfactant secretion by the cell, which is very important to survive after this injury. So this is also one of the key points in which mesenchymal stem cells, by the transfer of mitochondria, are supporting tissue maintenance. Other great work in which I found very interesting data is the one that was led by Emery Rodriguez, work with Jiren Asel and Jennifer Lebu, and a great team, that show that when you have mesenchymal stem cells and you activate platelets, these mesenchymal stem cells receive these mitochondria, and they tested differently. It's an amazing person, very compelling as well, that when they tested in the wounds, they observed that, for example, they had they have an effect with they add the mesenchymal stem cells, but when they use the MSCs with the activated platelets, they observe a better healing percentage, as you can see here. So this is very important and was a very good proof of concept that mesenchymal stem cells, when they go to a tissue, maybe we can potentiate their capacities and they can induce a better regenerative response. So this is key. Then in another context of mitochondrial transfer, because mitochondrial transfer has so many aspects, ones that, for example, mesenchymal stem cells use it for regeneration. The other one is that mesenchymal stem cells or other cells could be susceptible to be transferred by cells, like to transfer mitochondria and then induce better effects. And then, for example, in this case, if we don't get rid of dysfunctional mitochondria, especially when you have it close to the heart, you can induce dysfunction. So in this case, in a work made by Jose Nicolas Avila and with Sandra Martinez Salamanca, they observed that, for example, this very active tissue, which is the heart tissue, cardiomyocytes, they have to get rid of this mitochondria, and the macrophages that are around, they need to take this mitochondria to clear it and to make this tissue work better. So that is very important. And what they observed is that when they hampered this process, or when they depleted these macrophages around the heart, and they didn't uptake mitochondria, they observed that this unhealthy mitochondria was accumulating, which caused problems in the heart. Actually, the hemodynamics, for example, the diastole, systole, was less better in these cases. So this was very interesting. And then we have Jonathan Breston and Claire Crew work, in which they observed that adipose tissue is transferring adipose cells, are transferring mitochondria to the macrophages. And when you have a high-fat diet, or you are under obesity, well, not you, but in the model, you have a high long-chain fatty acid diet, these adipocytes are unable to transfer the mitochondria to these macrophages. The macrophages kind of like they don't uptake these mitochondria anymore. And so you have a lot of mitochondria circulating, one specific time mitochondria circulating, and they go to the heart. And as observed by Claire Crew, these mitochondria, as you can see here, that is being liberated, somehow induces antioxidant better capacity in the heart tissue. So it's very interesting, because in one context, you have mitochondria that's been getting rid of the cardiac tissue, as I showed you before, and then in this context, you have the mitochondria that it needs to come from this adipose tissue, for example, induce more resistance capacity in the heart tissue to resist, for example, Ross stress. So this is very interesting. So it's very puzzling how in the same body we can have this, in this model in which it's like a puzzle in which we try to have a coordinated idea of what could happening. We have these different aspects taking place. So it's very interesting to see that. And the quantity of articles that show that mitochondria transfer from mesenchymal stem cells by this horizontal mitochondrial transfer process is amazing. So there are a lot of articles. So Swati Paliwal made a very nice compiling review in the Journal of Biomedical Science, in which she listed all the some of the articles that showed that mitochondria is being transferred to other cells, and the specific process in which they are having a good outcome. So this is very interesting. So the key highlights is that so far horizontal mitochondrial transfer from disease induces immunomodulation. They, as shown in this and many others, they have the capacity to protect lung tissue and improve wound healing when they are transferred to other cells. And they have a metabolic, and they help in the metabolic and phenotypic reprogramming of other cells. So this was very important, and that's some of the things that we are that we were inspired to work here at the USFQ to move forward on that. So just a conflict of interest statement. I'm a scientific advisor of a company. It's called Lubitix. I'm the team leader of the Dragon Biomed initiative here at USFQ, more academic than entrepreneur now. I'm the CSO of Omnions, is a wellness biomarkers company. So what we intend to understand at the USFQ laboratory is that tackling mitochondrial dysfunction by mitochondrial transfer could help us to diminish or decrease somehow these hallmarks of aging. And this is also a very interesting idea that we developed all together with Keshev, that part of the hallmarks of aging, there is mitochondrial transfer, and that by mitochondrial transfer, we could be able to rescue some of these cells, but we still have like these missing links that I will discuss you after. That is this a process that is taking place when our tissues are in danger for prevention, or is also something that is hampered by aging? So these are key things that we need to work on. As Steffi mentioned earlier, there are different mechanisms of mitochondrial transfer, TNTs, microvesicles, for example. And we've worked since I arrived here in Ecuador in 2016, and now from 2020, we develop our resources to make this work. And everything was started by this by our work in France, an amazing team over there, and that I really thankful because they initiate my my way in the mitochondrial passion environment. I could say that because I was not so interested in mitochondria when I was starting this part. But we developed a tool in which we could transfer mitochondria, isolating it from secondary stem cells and put it in other cells. In this case, was cancer cells, and we use to induce different types of phenotypes. For example, increase oxygen consumption rate, decrease glycolysis, observing that, for example, cells that receive mitochondria, as many others, increase the ATP production, the cells tend to move more and to proliferate more. And we developed this patent that is now being used by many scientists all around the world. But when I arrived to Ecuador, we aim to answer what if mitochondria that is being transferred by a mix of different cells could actually activate the immune response, or could help us to maintain the T cells and all the leukocytes in a sample in a good state. So we wanted to see if they could also repair damage. So we have these PMCs, we expose them to UVC light, we cause harm, and then we use mitosections to see what happened. And we develop modifications to the regular mitosection protocol, in which we now adapted to non-attached cells, in which we have the non-attached cells and we transfer the mitochondria. And we observed that CD3 lymphocytes and CD14 monocytes have different patterns of uptake of mitochondria. So if we put it in the context that there are mitochondria that are free in, for example, in persons that are in good health or are, have overweight, this pattern could change. So the influence of the mitochondrial transfer in the immune cells could be huge. And as shown by Steffi, that this could be and mediate many different aspects, even the metabolic exhaustion of the T cells. So this could be very interesting to discuss after. So what happened is, to make a long story short, when we transfer mitochondria, we help them to recover their metabolic activity measured by MTT, as you can observe here. Like here's the induction of the 3 minutes UVR exposure. So metabolic activity was harmed, and then we recover it. This happens after the 24 hours. And reduction of cell mortality, which is enormous, is like when we use it at 3 minutes, and here we take a look after 24 hours, the quantity of cells that arrive to survive after the UVC exposure after receiving the mitochondria was a lot. And here we can observe that there were a lot of cells that actually survive the stress. And we published this with an amazing team. We have Maroon from Chile, we have Kristen Jorgensen from France. So it's we made this all together, and it was very nice because it was one of the first work experimental work here at the university regarding the mitochondrial transfer and also cell culture. So as I mentioned before, we collaborate with Patricia, and we observed that transferring mitochondria has an immunoregulatory potential. And so MTT changes the metabolism, induces resistance to UVR stress, and immunoregulatory profile. So make us think this could be very interesting to observe in skin, because skin is always exposed to UVR radiation and needs to constantly proliferate. So we all work together with the University of Los Andes, the Air Force Office of Scientific Research in the United States, LUBIGIX, and the University of San Francisco de Quito, in which we developed this system of co-culture in which we wanted to observe the mitochondrial network axis. So we transferred these mitochondria from the melanocytes to melanocytes, for example, and very interestingly, that these melanocytes didn't obtain mitochondria, so melanocyte doesn't transfer mitochondria quite well, and even when we exposed them to UVR, they were not receiving these mitochondria, or it's like transferring these mitochondria, which is good, which I don't know which is good, but we observed this process. We put, for example, different cells combined, and we didn't observe the mitochondrial transfer. But we put the melanocytes together with the keratinocytes, we observed a lot of transfer of these mitochondria. So, and even when we expose them to UVR, we observe an increase in this process. So, for example, here are the melanocytes and keratinocytes in green, melanocytes in red, without exposure of to UVR, and we observe that there is evidence of the transfer of mitochondria. But when we expose them to UVR, we observed that there was an increase in the uptake of mitochondria and the transfer of mitochondria from melanocytes to these cells. We made also the combinations and in between different cells, for example, fibroblasts to melanocytes, and we were able to observe there are some cells that didn't receive this mitochondria, which is good as using the same settings for all the cells, but observing that in one cases they were receiving mitochondria and in another case they were not transferring mitochondria is very important to dig a little more in the system. And this is the first work that show this transfer in skin. Then we wanted to observe if by separating the cells, there was changes in this transfer process. And what we observed is by when we put them division between them, we observed that the mitochondria lowered, the mitochondria was transferred lower in the proportion when they were separated. And then when we expose this transfer to UVR, there was an increase of this mitochondrial transfer, as you can see here, but not as much as the direct transfer between them. So we wanted to use fibroblasts as they seem to be more fragile when they are exposed to UVR radiation in normal skin conditions. So together with Steffi, we characterized these mitochondria that was transferred. We observed that they were fit, they produce metabolic, they were metabolically active, and we transferred them to this fibroblast, and we observe here that there was a reduction of ROS production in the cells that receive the mitochondria, more specifically to a specific concentration, and that the transfer of mitochondria from a single stem cell didn't induce ROS when they were transferred in these conditions. They induce, as you can see here, in different settings, and the proliferation of the cells that receive this mitochondria. And then by validating all these things in vitro, we pass through the in vivo model with the help of amazing partners and scientists on this part. We add the mitochondria to the wound, as you can observe here. And we were able to observe in this first set of data that transferring the mitochondria induces better wound healing at after 48 hours. And that, as observed by Anne-Marie Rodriguez, mesenchymal stem cells has also improved the wound healing of the skin, and we are performing many other assays. We are regarding FGF and many other proteins on these settings. And we wanted to later after to use these mitochondria to improve wound healing, but also maybe something protected to this radiation that here, particularly in Ecuador, we are very exposed to. But we know that, for example, one key aspect is to preserve this mitochondrial function, and we were developing a solution that could help us out to preserve this mitochondria. And we compare fresh mitochondria to the preserved mito, and by using different experimental settings, we wanted to compare if mitochondria in fresh media compared to the preserved mitochondria were able to induce the same proliferation, for example, as we can observe here. Our media of preserved mito was working well. They preserved the metabolic activity, or they induced the metabolic activity of the recipient cells, and we didn't observe too much DNA leakage in the solution. So so far, we are eager to continue working and to develop these great networking connections together with Steffi, together with Patty, together with the team in France. You all want to make more great science. We are thankful to our stakeholders, to Universidad de Concepción, especially to Steffi, to the people in Universidad de los Andes in Montpellier, to all our partners that help us out to move forward on this. And now some concluding remarks. How about that, Steffi? I think that we are kind of in time.
Yes, go ahead.
Excellent. So I think that pretty much we all agree that mitochondria have a dual role. This is a dynamic process, and it helps to restore cell function, induces immune regulation, for example, but it also is contributing to pathological processes related to, for example, cancer. And that further research is essential to boost the therapeutic applications, but we need to move cautiously. So for future directions, for example, we need to dig more in the molecular mechanisms related to this mitochondrial transfer. Mitochondrial variations, as we need to understand more, is like the disposal of mitochondria that is not working well is good for the tissue. So we need to improve the clearance of these unhealthy mitochondria. But it's also, what if some mitochondria, more specifically, for example, from adipose tissue or other donors, are improving cellular function? So we need to understand that. And of course, the therapeutic conscience that HNT, of course, seems to be regenerative and reparative, but we need to understand more the risk that cancer cells in the same tissue could also acquire this transfer mitochondria and induce, for example, as you showed early, Steffi, cell exhaustion. So yeah, thank you very much. And let's move forward to this. I will stop sharing my screen and let's move forward to the questions.
Well, thank you very much
Thank you.
for your excellent presentation. And we are already over 1 hour, but I mean, if it's fine with the audience, because the topic is so interesting, maybe we still can ask a few questions. Or are we being cut off by Zoom, David? Do we still have some time?
There's no problem.
Okay, so let's continue a little bit, maybe. I mean, one question, I mean, we know a lot about the mitochondrial transfer between cells, about the mechanism of action. But you didn't say anything about the mechanism, how isolated mitochondria are being taken up by cells. So my question is, what is known about this? And as far as I remember, this was already observed in 2006, 2007, that isolated mitochondria are being taken up by cells. So what's known about the mechanism? If there's nothing known, fine. I mean, don't. But I'm very interested in this.
Well, I've seen in some studies there have been shown that the activation of one cell induced the formation of the nanotubule between two cells. So it's not that just one cell create the nanotube. Both cells require
No. As you introduced, you isolate mitochondria.
Yeah.
You wash them, you treat them, whatever, and then you add them to cells. And these isolated mitochondria are being taken up. There are no nanotubes involved.
Ah, you mean in the lab, not in the... not physiologically.
Yeah, no, because in the end, I mean, we have many questions aiming at this. For example, one question is, how long will it take and what is needed for artificial mitochondrial transfer to reach a commercial level and be available in pharmacies? And as you also well know, there are even startup companies out there who mass produce or who claim to mass produce mitochondria and then take them as a medicine, as a therapy. So it's bit, yeah, okay.
No, I think that there are many. It depends on the cell, and if we, for example, expose different immune cells to the mitochondrial transfer, different cells will have patterns of uptake. So this is what we observe, and this is what also has been observed by Patty. There are some mechanisms, for example, heparan sulfate present in the outer membrane. There is micropinocytosis. There are actin-dependent mechanisms of uptake of in internalization of mitochondria. However, mitochondria that is liberated and mitochondria that is isolated is super different from one cell to another. So
yes,
we really
exactly.
need to understand more. It's like we need to focus in that mitochondrial transfer axis between one specific cells and go deeper on that part, because one outcome could not be the same of other systems. So that is super important.
Yeah, exactly. I even think the field should be split between intercellular mitochondrial transfer and mitochondrial cellular uptake of exogenous mitochondria. And the big hope for future pharmacies is for uptake of, well, put mitochondria in a bottle and give it to somebody. Okay, Marvin, maybe if you want to go ahead, pick questions.
Yes. Thanks again for Andres and for Stiffy. So I think we have other question. The answer can be quickly, or maybe you can answer later. So if we go to the first question, how long will it take? What is needed for artificial mitochondria? The first question, I think, by Isaac Escobar. Andres or Stiffy, very quick, because we have other questions really.
Yeah, it's about my deception, Andres. I don't know if they want to see if my deception can go to the clinic, I think, right?
Yeah, I would like to see that as well, but it's still risky. I don't know if infusing myself still with mitochondria would be helpful. I don't know. But okay, so mitochondria can be uptaken in around 4 hours. And we've seen that, for example, in 20 minutes, they're already starting to uptake it, but in 4 hours, it seems that they arrive to a plateau. But then these cells, for example, T cells, in how long they're going to preserve this mitochondria, it seems that takes 72 hours depending on their capacity of proliferation so far. But for example, if we mitocept MCs, they stay a little longer, but it all depends on the capacity of their proliferation.
Okay. Question number 2 for Stiffy. It is any evidence for the molecular machinery of mitochondrial transfer?
We could reverse tumor progression. Yeah. That's a difficult question, because then it has to be more research about specific mechanism of mitochondrial transfer in tumor that is not present in healthy tissue, which I think we don't have that so far. We don't have a specific mechanism that only requires proteins from tumor. Therefore, if you block any of the transfer mechanism, you're gonna affect like a normal mechanism for healthy tissue as well. I think that's very tricky. It would be very important and very clever to find one module.
Maybe we can select this question and we can comment them after the webinar. How does mitochondria donor cells maintain homeostasis after donating the mitochondria to other cells?
That's depend on the quality of the mitochondria, because if your mitochondria is damaged, you release that, it's better for you. If your mitochondria it's in a healthy state and you lose it, you're going to lose function.
Okay, I
Yeah,
think
and there's also one interesting work by Anne-Marie Rodriguez in which she observed that mitochondria that are receiving damaged mitochondria, as like MSCs that are receiving damaged mitochondria, they induce them to make more mitochondria and to liberate more. So that was a very nice work.
I think maybe the other question we can answer, the last question, what is the red line we must not cross when manipulating or inducing mitochondrial transfer, whether in cancer or in longevity, regeneration? I think this is, the conclusion question for both of you. On the other one, we will send you all the question, and if you wish to answer, we can send to all participants.
We will open a forum.
Yeah.
Very quick
There's a lot
comments, the bad or the good.
I think as like John recently established nomenclature, probably it's important to define function, that a criteria for function of mitochondria to be like a good mitochondria to be transferred. I don't know if that is defined. I don't know, Andres, if you know that a mitochondria had a checklist to see, okay, this mitochondria can be good for therapy compared to other mitochondria.
Yeah, well, it comes to my mind after receiving a lot of reviews, but I think that, well, establishing their activity. Mm-hmm. But we now know that it depends on the goal. We could use mitochondria that is not too well in order to induce a specific response in the recipient cell. So everything will depend on the use and what the authors will reivindicate. It's like, for example, it is highly important that mitochondria will be in a good respiratory activity when you are seeing that it's being introduced into the heart, for example. And also macrophages around the heart will uptake this mitochondria. So what this would imply in that sense. So there is a need for guidelines when mitochondria will come to the to these clinical trials. However, there is an absence of regulatory pronunciation, statement regarding the use of mitochondria in therapy. So there is a growing field. It's a growing field, and most of the time is like as science move forward, and there are amazing companies already in the field. I think they are doing a good job in trying to develop the best mitochondria for specific purposes. For example, Minovia is using mitochondria isolated from placental or embryonic tissues to develop models to treat the person syndrome, for example. So they are transferring this mitochondria to hematopoietic stem cells, and they are using it to reprogram them. And it's very interesting that these healthy mitochondria, without harming mutations, it seems, are staying in this lineage, and they're using therapeutic purposes. So there are a lot of things in which we should work on, and I'm happy that there are a lot of movement and very interested of a lot of people now in these things.
Thank you very much. Thank you. Wortmar, it's yours. I think that we don't have enough time to answer all the questions. Maybe we can, answer them by email. So please.
Yes. So again, I mean, I would like to thank you again for this excellent, highly interesting presentation. And I'm looking forward to meeting you in Berlin in October. And having said this, I mean, I think I would like to close our webinar. So thank you again, and thank you for everyone having attended it. And again, the questions will be forwarded to both presenters and yeah, no beer, yeah. Will to both presenters, and you will get a reply from the presenters. Okay, so Andre just showed 2 glasses of beer because I mentioned Berlin, Germany. Okay, so
and it's October.
In Oktoberfest, yeah. Okay, so again, thank you very much, and that's the end of the webinar. Thank you.
Thank you, Marwan. Thank you, Marvin. Thank you, Stefan.